Hello, everyone, and welcome to our conversation on transformation. So, just a reminder that transformation is a form of horizontal gene transfer in which a bacterium is actually going to take up free DNA from the environment. So, check out this picture over here on the right. And what I want you to notice is here's the bacteria, and here is that free DNA. And so that cell is able to take up the free DNA and integrate it into its own chromosome. Okay, so notice there's no cell to cell contact, there's no phage, nothing, no other microbe needs to be involved. Okay, now there are some kind of picky requirements that need to be met for this whole process to actually occur successfully. But for the moment, I want you to just notice and make note that this isn't a bacteria that is able to pick up DNA from the environment. All right, so let's talk a little bit more about this DNA. So sometimes this DNA is called naked DNA because it is not carried inside another cell or virus or anything like that. Okay, so as I mentioned, unlike conjugation, transformation does not require cell to cell contact. Okay, so how does this happen? When is there just DNA hanging out in the environment? Well, when other bacterial cells die and break apart or lyse, fragments of their chromosomes and plasmids are released, right? If a bacteria bursts open, all of its insides, including the DNA, are released and available to potentially be picked up. But not every cell or bacteria is capable of picking up this DNA because DNA is rather large, even when it's been blown to bits, and it's charged. And so it doesn't easily cross that plasma membrane. So only cells that are said to be competent, so let's write that word down. So only competent, my pen is going a little wonky, sorry y'all. Competent. Only competent cells are actually able to bind and bring this external DNA into the cell. So, some bacteria are naturally competent under specific environmental conditions. If the DNA enters the cell and is maintained either by joining the chromosome or remaining as a plasmid, the recipient cell may express new genes and gain new characteristics. So, transformation is actually a process that scientists do in the lab a lot. It's a really important laboratory tool where we can make certain bacteria competent. We then expose them to DNA containing whatever gene we're trying to study, and kind of force them to take the DNA into its cell so it can then express it and we can study that protein. So, for example, if we wanted a bacteria to carry an antibiotic resistant gene or produce a particular protein of interest, um, we can do something called like a heat shock where we kind of heat them. And then I think there's them going, ah, but as they go, ah, they take up the DNA. So it's pretty cool, and it's not a terribly difficult process either, but it was a game changer in the research realm. All right, so how did this come about? How did we learn about transformation and even horizontal gene transfer as a process? So our understanding of transformation began with the work done by a scientist named Frederick Griffith. So again, on your exams, I'll just call him Griffith. And he was studying Streptococcus pneumoniae, which causes pneumonia. And he had two different strains of this bacteria. One had that capsule, remember that sticky structure that helps evade the immune response, and that was called the S strain. It was virulent, it killed the mice. Okay, it's a pathogen. The other strain or version of this bacteria was called the R strain, which stood for rough because it did not have the capsule. So, not having the capsule made it not dangerous. It was non virulent. The mice were not killed when exposed to this R bacteria. Okay, so let's look at the picture just to make sure um, that we understand what's happening. So we had our our control setup where the mouse was exposed to the R strain, which was non-virulent. It lived happy go lucky mouse, no problemo. But then what they did is they took the smooth strain, which was virulent, and heat killed them. Okay, so that means expose them to heat in order to kill them. And so when those virulent bacteria were exposed to heat, they died. So when those dead cells were put into the mouse was fine, right? Because the dangerous bacteria had been killed. So those two made sense. Okay, here comes the interesting part. When Griffith mixed heat killed S strain bacteria, okay, so killed the dangerous ones, um, excuse me, and then he injected those cells into the mice. Excuse me, I'm sorry, I got ahead of myself. So he took the rough strain, the R strain, the blue ones here, that are known to not kill the mice, as we saw in the control experiment. And then he mixed that one with the heat killed S strain. We expected the mice to live, right? Just like what happened in control in experiment one. But that's not what happened. The mice, sorry if I could speak, the mice died. Okay. What was even more surprising is he was able to recover living S bacteria from those mice. Okay. And so Griffith said, all right, well, I guess something from the dead S cells had gotten into those safe living R cells and made them virulent. Okay. And so he actually at the time referred to this mystery substance as the transforming principle. At this time in 1928, he did not know that DNA was what was happening. But this finding that the safe R strain, when mixed with dead S strain, that R strain was picking up pieces of DNA from the dead S cells and it was transforming, hence the name, transforming those R strain cells into pathogenic S strain cells. Isn't that snazzy, y'all? I think it's super snazzy. All right, so essentially, take home message from that was that the DNA was released from the heat killed smooth strain and was taken up by the living non virulent R strain, and it gave them the genes they needed to cause disease. All right, y'all, that is it for our conversation on transformation. And as always, let me know if you have any questions. Bye, y'all.